It's my pleasure to introduce Paheli. She was an undergraduate, she had an undergraduate degree at UC San Diego in chemistry, actually, and she worked in science concentration in chemistry. And she worked in the lab of Henriette Barbanel in the physics department, doing work related to nonlinear systems. Sorry. Work related to nonlinear systems and to neurons and neuroscience. And that sort of shows her broad background in the sciences across chemistry, biology, and a lot of math. And then I was fortunate for her to come to my biomath department in computational medicine now at UCLA and be in my group, where she's taken sort of interest that I've had for a long time about branching networks and optimization, and applying those same ideas to neurons, which is it's a really cool thing that no one's ever done in my group before, which involves basically image processing, image analysis, a lot of theory on branching structures and the dynamics and flow through neural networks and how to optimize in different ways and how that's compare and contrast to other systems that have been looked at before. But that's what she's going to tell you about now, so I'll let her do it. So take it away, Paheli.
Can everyone see my screen?
Yeah.
Okay.
But we see, okay.
Okay.
Great.
Okay, thank you. Thanks for the introduction. So what are neurons? Neurons are the basic building blocks of the brain and the nervous system. And they have these centralized cell bodies and these branching processes that connect them to one another, and they transmit information in the form of electrical signals and chemical signals. And these connections are very important. They allow us to transmit information from the brain to the body and from the body to the brain. And they allow us to perceive our environment and also respond to our environment. So they're the basics of information processing and communication in animals. And if we zoom in on one particular neuron, we can see that there are two main types of processes. So they're axons, which generally conduct signals from the cell body to the synapses, where they connect to the processes of other neurons. And at the synapses, they connect to dendrites from other neurons, and these dendrites generally take signals from the synapse to the cell body. And even within axons and dendrites, we can see these differences in morphology in this image here. So the axons generally have these very long parent branches, and dendrites have these shorter branches that shorter branches that branch earlier and have more extensive trees. And so this is within a single cell. But if we look at a lot of different cells, there's a great amount of diversity in the axons and dendrites. So there's some processes that are just micrometers long, and then there are other axons that extend over several meters. And within the dendritic trees, we have a lot of different types of morphologies. So in this picture near the letter A, that's a Purkinje cell, which is a classic example of a neuron that has projections that are in a two-dimensional plane, and there are others that project into three-dimensional space. So we can see this diversity in structure. And in the past, in order to differentiate between these different types of cells, we relied on these visual qualitative features. But recently, because of technologies that allow us to measure the processes individual radius and length, we're able to distinguish between these using more sophisticated quantitative metrics. But still so we have this diversity in structural form, but there's also a great diversity in the function of these different types of cells. And one of the important goals of cell type classification in neuroscience is to develop this kind of correspondence between structure and function. And so the way that we've approached this is to use biological scaling theory. So we use biological scaling theory to understand how in different biological resource distribution networks, such as the cardiovascular system, we want to understand the connection between these branching patterns and function. And we do this by thinking about how these branching patterns might be optimized based on evolutionary selection pressures and to optimize a certain function. And when we look at the structure of the cardiovascular system, we can see the centralized heart and then the extending branching vessels. And there are some structural similarities between this and neurons, where we have the centralized cell body and then the branching processes. And so based on these structural similarities, we thought that it might be useful to work off of the mathematical theory we've already developed for cardiovascular networks and apply it to see if we can determine some connections between structure and function for neurons. So what do we mean by scaling? Well, we're interested in these quantities, which are basically looking at the relative radius and length. So instead of looking at these absolute quantities of radius and length, we look at this branching network and we want to see the comparison between the radius of the daughter and the radius of the parent, and the length of the daughter and the length of the parent. And we can come up with predictions for these values based on function and optimizing a mathematical function that relates to some kind of biological function that we're optimizing. And so if we think about neurons, one way that I like to think about this is sometimes you might think about when you're in the kitchen, you accidentally touch a stove and it's hot, so you want to move your hand really quickly. And so you might imagine that this biological process involves very rapid conduction of information. And so one function that the structure might be designed to optimize is minimizing the conduction time delay so that you're able to transmit this information very quickly. And another function we might think about is that, so transmitting this information takes a lot of energy, but there are only finite resources available to the brain. So we might think that the structure might also be optimized in order to minimize the energy consumption. So based on this, we come up with a general objective function that we want to minimize. So in equation 1, the first term relates to energy, which we've represented as power loss due to dissipation. And we formulated this based on the fact that we think about these processes as wires and through which there's an electrical current. So we formulate this as resistance to current flow through a wire. And then this term in the denominator where we have n to the power of k, the little n is the branching ratio. So in this case, we have a bifurcating network. And so for a bifurcating network, that branching ratio is 2. If, let's say, at the branch, we had 3 daughters, they would be equal to 3. And as you can see at each level, the number of branches is equal to the branching ratio to the power of the level of the network, so k. So that's where that term comes from. The second term has to do with conduction time delay. So the term in the numerator is the length of each branch, and then in the denominator, we have the velocity. And this parameter epsilon has to do with myelination, which is, for neurons, we have this insulation that surrounds the fiber and facilitates the conduction. And so previous results have shown that for unmyelinated fibers, the velocity is proportional to the square root of the radius. And for myelinated fibers, the velocity is proportional to the radius. So we think about varying this parameter epsilon. So for the unmyelinated case, it's equal to 0. And the myelinated case, it's equal to 1/2. And we're summing these individual values over each of the generations. And then in the third term, we have in equation 1, we've just shown an arbitrary representation of our constraint functions. So these are the biological quantities that we want to hold constant. So the first two terms we're minimizing, and the third one, we're holding these to be fixed. So equation 1 is the general case, and then we want to think about some specific cases. So we might vary the parameter alpha. So if you set alpha equal to 0, the first term disappears. And so we only have the second term. So this is a case where we're only minimizing time. And an example of this is in equation 2. Another case would be where we set the parameter alpha to be equal to 1. So the second term disappears and we only have the first term. So in this case, we're minimizing only power. And equations 3 and 4 are examples of that. And then when we consider the functions, the constraint functions that we want to hold to be fixed, in equations 2 and 3, we're considering 3 constraints. So volume, size, which we've represented as the mass of the cell, and lastly, space filling, which is the fact that we are optimizing this network in order to fill as much of the space as possible. And this could be either 2-dimensional space, so a plane, or 3-dimensional space. And the function 4 is very similar to the third one, except instead of having the volume constraint, we've included time delay as a constraint. So we're holding time delay to be fixed in this case. And so from these objective functions, we use the method of Lagrange multipliers to solve for optimal values of the radius scaling ratios. And so we consider 4 main cases. So first we consider time minimization, which is shown in equation 2, and we consider the unmyelinated case where epsilon is equal to 0, and the myelinated case where epsilon is equal to 1/2. And then we also consider power minimization in equations 3 and 4, where we consider the case where we hold volume to be fixed in equation 3, and we hold time delay to be fixed in equation 4. So these are the results of the theoretical predictions for time minimization, with the myelinated case, the unmyelinated case in the left column, and the myelinated case in the right column. And then here we have the results for power minimization with the fixed volume case in the left column and the fixed time delay in the right column. And one thing you might notice is that we did vary the dimension of space filling here, but we found that the dimension of space filling didn't affect theoretical results. So as you can see, they're the same for both cases for radius scaling ratios. And we find that the dimension actually does affect the theoretical predictions for length. But in this analysis, we focused on radius. So I'll come back to that later. But just to let you know that we did consider this, but it didn't affect the predictions for radius. So now we wanted to compare these theoretical predictions to data. So I got this data from a public online database called neuromorpho.org. And here it has a range of reconstruction from neuron images from a range of different species and cell types. So the species that we use range from insects and rodents, and then they range all the way up to elephants and whales. And you might notice that the axon data, there's less of it, and this has to do with the resolution limit, because often the axons are very thin compared to dendrites. But given the differences in morphology that you see in the images, I thought it would be useful to kind of compare the results for these 2 different types of processes. So as you can see in the results, the mean scaling ratio for radius in dendrites is closest to the cases where you're minimizing power, and the mean scaling ratio for axons is closest to the cases where you're minimizing time, and particularly the myelinated case, which makes sense because based on what we know about the biology, we generally associate myelination with axons and not with dendrites. And as you can see for the dendrites, the mean lies in between the two predictions for minimizing power, but it's not particularly close to either one. And this might be due to the fact that this data combines many different cell types. So what I wanted to do is kind of look at some of these cell types individually. So I chose to look at Purkinje cells and motoneurons initially because of the fact that Purkinje cells are a classic example of neurons that project only in two-dimensional space, and motoneurons project in three-dimensional space. So I wanted to compare and see if this had an effect on the scaling ratios. But in our theoretical predictions, we found that the dimension of space filling didn't actually affect the predictions. But we do see this difference in the means in the data. So I propose that this difference was actually not due to the dimension, but rather due to differences in functional roles. So here we find that the Purkinje radius scaling ratio mean is closest to the predictions for power minimization with fixed volume, and the mean for motoneurons is closest to the function for power minimization with fixed time delay. And this suggests that time plays a greater role in constraining the function of motoneurons. And this makes sense based on what we know about the biology, because the Purkinje cells are in the the cerebellum, which is a brain region that's associated with voluntary motor control. And motoneurons are in the spinal cord, and they connect to sensory neurons and also connect directly to muscles and are part of the sensory motor system, which is often associated with very rapid reflexive movements and responses to the environment. But in order to test this importance of time even further, I wanted to look at scaling ratios in the peripheral nervous system neurons themselves. So I looked at this and I found that the mean for peripheral nervous system actually was closest to the theoretical prediction, minimizing time in the unmyelinated case, which makes sense because we're focusing on dendrites and not axons. In addition to that, we also, when we looked at this particular function where we're minimizing power and we have this time delay as a constraint, we're able to derive a functional scaling relationship between conduction time and mass of the animal. So the way we do this is we represent this power term as capital P and this time term as capital T, and then we find the stationary point. We take the derivative with respect to radius and length and rearrange a few terms, and then we get this form in the second line. And then based on previous results that have shown a proportional relationship between the mass of a cell and the fourth root of the body mass of an animal, we made some substitutions, and then we varied this function with respect to the mass of the animal and solve this differential equation. And we end up with this equation that relates conduction time to the mass of the animal. And in this case, B is a constant that's experimentally determined. That's the proportionality constant between energetic cost and mass of an animal. And so here we find two terms. One of them has a 1/4 power and the other one is linear. And when we look at experimental data, so we look at data comparing conduction time delay and body mass. And these species are ranging from shrews to elephants. And we find this, the slope of this log-log plot suggests that the time delay is proportional to this body mass to the power 0.3. And this is close to the 1/4 power, which is 0.25. So this suggests that these experimental results support the theoretical results that we find where we find this 1/4 power law. So in conclusion, our results suggest, based on our comparison of the theoretical predictions and the data, we find that axon structure seems to be governed by time minimization and myelination, and dendrite structure is governed by power minimization. And within dendrites, we looked at two different types. So Purkinje cell dendrites are governed by power minimization with fixed volume. Motoneuron dendrites are governed by power minimization with fixed time delay. And then we looked at peripheral nervous system dendrites, which are governed by time minimization without myelination. And then both in our theoretical results and looking at experimental data, where we find this 1/4 power law relationship between conduction, time delay, and body mass. And this is something that is interesting in the context of evolution also, because we find that for larger animals, there's a greater time delay. And this could also relate to the emergence of hemispheric specialization in larger animals. So because of the longer time delays, it might make sense to cluster cells that encode similar memories and skills closer to each other. So some of the next steps that we want to take with this. So as I mentioned, we focused on radius scaling ratios in this analysis. And we did actually look at length. What we found, so in the radius scaling ratio distributions, as you might see, we find these normal distributions. And for length, we actually find this exponential distribution. But the results don't compare very well to what we see in the data. But we've recently, for cardiovascular networks, we've been applying different methods of labeling the branching generations. And we've found better results. So that's something that we're looking into applying and learning more about the role of the length scaling ratios. And another thing is, so I'm gonna go back to here. So in this whole analysis, we've made the assumption that the radius is symmetric in this branching. So the radius of both of the daughter branches are equal to each other. And this is often not the case in biological systems. And previously in other biological systems, we've looked at asymmetry of radius and length. And so in this case, we're assuming that because the radius of the two daughters are the same, they have the same relative ratio with the parent. But if you consider different radiuses, you might find that there are two different scaling ratios, and we can calculate parameters such as the average between those and the difference between them. And we've actually been able to use these parameters as features for classifying between different types of networks. So one thing that we're interested in looking at is applying these methods to also classify between different types of neurons. And the exciting thing about this is because these parameters actually have functional significance. So using these parameters to classify would not only tell us about the differences in structure, but also the differences in the basis of like the functional basis behind those differences in structure. So I just want to acknowledge QCBio for providing this platform, and my advisor, Van Savage, and Alex Brummer, a postdoc who works in our lab, who has contributed a lot to this project, and all the members of the Savage Lab, and then the NIH and NSF for supporting our research. And also, these are the references I used for theoretical formulation of the model. And then these are the published results, and I used the experimental data that was put on the online database from these papers. So these are for Golgi and Purkinje cells. These are for motoneurons, and these were for axons and PNS neurons. So are there any questions?